Okay. Well, we are all almost here. It's so good to see your faces. I want to welcome you all to this day in January of 2021, and just so thrilled that we are meeting this week when our spirits are lifted, our hearts are filled with hope. We've had 4 years of hardship that has been stored up in various ways, including in our bodies. We have been witnessing the fragility of democracy, the fragility of life. We have a really exciting program today. We're first going to have some announcements on meetings, memberships, and members. So let me start with members. Some of you may have already heard that on December 4th, we lost Mark Laudenslager to the complications of COVID. Mark was a beloved ABMR member, collaborator, friend. His obituary from University of Denver and Memorial Fund are on our members page. We have also lost Kenneth Walston in October, a distinguished researcher in the nursing school at Vanderbilt, very dedicated long-term member to our academy. In the last year, we lost other phenomenal members, Ron Glazer, Bruce McEwen, Chuck Carver. We're going to take a moment of silence to remember them and anyone that you want us to acknowledge together. I'd like you to type their name in the chat. We have lost 400,000 Americans to COVID, but more importantly, we have lost 2 on our planet who have died from COVID this year. So let us take a moment of silence together. Okay, so today we're going to dive into the phenomenal connections between our social world, our mind, our immune response in terms of how we respond to infections, to vaccinations, and the psychology and sociology of vaccination acceptance. First, we just have some announcements from our council members. In terms of our annual meeting, the good news here is that most of you have signed up already. We've moved our reservations several times, and so it's been a lot of work for each one of us. Thank you, Elena, who my lab manager, who has been managing this, and Lori McBurney. So it's a complicated algorithm now that we're each in one room on site with a limited number of people on this resort campus. So many of you are on a wait list, and you've tried to move your room, but you can't yet. So what we have, because we have filled up the rooms that we've reserved. So what we've done is we've extended our reservation at the resort to February 15th, so that anyone who still wants to sign up, there are probably 15 people who have not yet moved their room. We have new members who I hope you will sign up. You can be off the wait list on February 15th when we extend our contract. So we need to commit financially to these extra rooms. Okay, so I'd like to turn it over to our president-elect, who will Anna Marsland.
Thanks, Alyssa, and good day, everyone. I think this is a wonderful week for optimism, and as an optimist, I am feeling really good about things at the moment and seeing the world in a whole new light. You know, this is fueled a little bit by many things, but it even looks like our numbers of cases here in Allegheny County, Pennsylvania, may be plateauing at this time too, which is encouraging. And as we begin to get back to normal, I am about to restart looking for somewhere for our June 2022 meeting when, fingers crossed, ABMR will be able to get back to, as we've done things in the past, and hold a normal meeting when we can all again be together and celebrate. I wish I had somewhere to announce. I had ideas before we got shut down. I am imagining our next meeting will be certainly on the East Coast, possibly in the Northeast. I have a couple of folks I'm talking to, but if anyone has places or ideas for a place where you think it would be good for us to meet, please email me and let me know. I am very open to input at this stage. I'll be working hard on this over the next couple of months, and by our meeting, I hope we'll have announcements and be able to tell you exactly where the June 2022 meeting will be, which is pretty exciting. I also just want to take a moment, we have a busy schedule, but I want to take a moment to encourage people to step up, to volunteer to join the leadership of ABMR. Being a leader in this organization has truly been a remarkable experience. I have been honored to rub shoulders with people who I hold in such high esteem. But it has also been a really small group that has been supportive in my developing leadership skills. And it's been fun. And as we move forward, I know in a second page is going to tell us about a number of openings that we have for people to join our executive committee. And I know many of the faces I see in front of me and on the other 3 pages have already, served ABMR and the Academy and served in leadership roles. But for those of you who haven't, I really would like to encourage you to step up and experience what is a really remarkable opportunity to be a voice in the future of our organization and the contributions that it makes, which I think are pretty remarkable to our field in these changing times. So it's just a plug we're going to be nominating. We have a number of openings coming up for leadership positions. I know we're all completely overwhelmed with service at multiple levels, but I do think that this is a really valuable thing to consider and would encourage people to step up. And also, please do let me know if you have a special place in the Northeast that you think would be a suitable venue for our June 2022 meeting. So good to see you all. And I'm going to hand over to Paige, who's going to tell us, I believe, about the opportunities that are available.
Thanks very much, Anna. And I just want to echo Anna's sentiments about serving ABMR. It has, it has really been a real pleasure to work with Alyssa during her tenure, extended tenure at that, as president, and the other members of the leadership that have really invested additional time in making sure that our academy remains functioning and progressive. And so I want to thank, Alyssa for her leadership, Anna for her leadership, and Annette and Viola and others in particular, and Martika, who have hung in there with us during this extended period of time. But life must transition, and we have witnessed a very successful and peaceful transition earlier this week, despite January 6th. And so with the decision to hold our next in-person annual meeting in October, it is time to hold elections so that we can honor the tradition of inducting new leaders into the society as leaders. So we have the president-elect position, the treasurer position, 4 EC seats to be filled, and then 2 membership committee at-large seats. I see that my dear friend Jerry Sulls is on, and he has served as one of the membership committee at-large seats. The other seat was occupied by our dearly departed Ken Walston. So those are the positions that are currently will be competed and available for our next election. So please look out in your emails for the nomination solicitation request and send those nominations in so that we can have an election. All right, thank you.
Hello, everyone. I like to think of the longer terms as extended disco versions for those who are in my cohort. It's truly my pleasure to announce our new ABMR members from the year 2020. They are in alphabetical order: Dr. Mary Carlson, University of Calgary; Dr. Bernard Chang, Columbia University Medical Center; Dr. Christopher Fagundes, Rice University; Dr. Carolyn Fang, Fox Chase Cancer Center; Dr. Cheryl Giscombe, the University of North Carolina at Chapel Hill; Dr. Jeanette Ikovics, Yale National University of Singapore; Dr. Chandra Reynolds, University of California, Riverside; and Dr. Kavita Vedhara, University of Nottingham, whose talk we're very much looking forward to today. A big congratulations to all of you. Our new members have been invited to give introductory talks during our in-person meeting this October, and we very much hope you can join us. And if you haven't already done so, please give Multiversity a holler about grabbing a room. Please also note that our 14 new members from 2019 will be giving their introductory talks at our June 18th virtual meeting on climate and behavioral medicine. Looking forward to all of it, and I yield the floor to Mustafa.
Thanks, Sherry. So nice to have you all here today. The next phase of this process is to nominate new fellows. So please consider nominating a colleague who you think would make outstanding contribution to the academy and have already made significant contribution to the field. And this can be usually demonstrated by widely cited papers that they have published, grants, and editorial board, or senior administrative roles that have significantly advanced the field of behavioral medicine. This year, the council has modified the process of nomination a bit, made it a little bit easier. So now the nomination package this year should include the nominee's CV and at least one letter of nomination from a member of the academy. The requirement used to be at least 2 letters. The deadline for submitting the nomination this year will be October 22nd, and that is 2 weeks after our annual meeting. So in coming months, we will send out more details and information about the nomination process. But in the interim, please feel free to reach out to me, to Sherry, or to any one of us in the council for any information. Thank you.
Thank you, Mustafa. Okay, so I want you to be prepared for what's next. We are going to have about an hour of presentations and discussion. And then we are trying something new, which is a new way to connect with each other by having 30 to 40 minutes of discussion and updates among all of us, among members. So please share something. It could be a new finding that you would like people to know of, a new program, a new initiative. It could be personal news. And we'll start that section with an update from John Ruiz, who is our new president of the Behavioral Medicine Research Council. So he'll update us on the council. Okay, so we are going to move into a very well thought out, well-planned section of short lectures. The background reading, very foundational papers and reviews are on the ABMR website at the very bottom of our meeting page. And so those are great resources for trainees, et cetera, and for us. And we're going to hear a more high level discussion of three areas of this. So let me tell you about our speakers. And we're just so pleased that Kavita, Steve, and Heidi are joining us because they could not be in more demand right now. Steve described it as a tsunami of demand. Kavita said her workload has increased threefold during the pandemic. And Heidi's work on the Vaccine Confidence Project is of course become of not national, but global concern. So Kavita Vedhara, our new member, is a professor of health psychology at the University of Nottingham. She is a pioneer in PNI. She started off studying HIV, chronic fatigue syndrome, and the psychosocial factors in infections and inflammations, and has turned to vaccination responses. She is really the leading researcher on understanding the malleable biobehavioral psychosocial factors influencing vaccination response. She's been part of her university's pandemic response. And she's also looking at short-term interventions that boost vaccination response. So we're all, whether we're vaccinated or not already, we're all going to pay close attention to that. Then we'll hear from Steve Cole, already well known to us all, professor of medicine and psychiatry at UCLA, who has been studying the biological pathways through which social environments influence gene expression by viral, cancer, and immune cell genomes. Steve is the father of the field of social genomics, and he's a very generous collaborator. He is also part of his university's COVID response. And also because of his expertise on loneliness, another pandemic epidemic, he has also been very busy. And then lastly, we're so pleased to host Heidi Larson, who is an anthropologist from the UK who's been studying the social, cultural, and psychological factors that determine vaccine hesitancy in a time of social media and misinformation. Misinformation is a public health crisis. So without further ado, I welcome Kavita.
Thank you very much. And let me start by wishing you all good morning, good afternoon, or good evening. I'm not quite sure which time zones everyone is in, so I hope that covers everyone. Firstly, let me start by saying what a huge privilege it is to be here today, not least because it is my first meeting, but also it's provided me with this opportunity to speak today with both Heidi and Steve. And the three of us have decided in the limited time that we've got available to us is what we'd like to do is to give you a broad brush overview of the psychological and behavioral exposures that are critical to our understanding of our relationships with viral infections and the effectiveness of vaccines. This was billed as a fireside chat. So we took that literally and we thought, okay, we will make this a little bit about science and it'll be a little bit autobiographical as well. So please bear with us. And our intention very deliberately is to take a historical perspective. And we thought this would be useful to kind of reflect back on where we have come from, because we hope it'll open up fertile discussion in terms of where we go next, particularly with the challenges that we all face with this pandemic. So that's kind of a little bit of background about our approach. In terms of what we're going to cover, well, what I'm going to start with right now is a little bit of history around the early research looking at psychological and behavioral influences on viral infections. I'll then turn to Steve, who's going to unpack some of the mechanistic data, particularly in the context of HIV infection. He'll pivot back to me briefly so that I can just review some of what we know about vaccine effectiveness in this context. And then last but not least, I'm going to turn to Heidi, who's going to talk about what I think many of us consider to be one of the most important ways in which psychological and behavioral factors influence our relationship with viral infections, which is in our decision to have a vaccination. So that's just a quick overview of what to expect. So let me start then with this issue of psychological and behavioral correlates of our responses to viral infections. And much of the work I'm going to talk about really started in the 80s and early 90s. And for those of you who are too young to know what was going on then, Let me tell you that that was the decade in which MTV was launched. It was the decade when the public was first introduced to mobile phones, which of course are now ubiquitous. And those of you who know me well will know that it was an important decade for me because it's when I first discovered George Michael. But that's about as autobiographical as I'm going to get. But in addition to these very important events, there was some really exciting behavioral science going on at the time in the context of viral infections. And with the benefit of hindsight, it appears to me that actually the field was almost adopting a bit of a pincer movement. So we had one area of work which was focused primarily on our relationship with latent viral infections. And here the trailblazers were Ron Glaser and Jan Kiekolt-Glaser. And then in parallel, we had the viral challenge studies. So here we're not so much looking at what happens when we've already got a viral infection, but what are the determinants of who is going to become infected when they're exposed to a disease. And of course, the pioneer here was Sheldon Cohen. So I'm going to briefly look at what we learned in those early days in both of those areas. So in thinking about latent viral infections, firstly, just to be clear, latent viral diseases are examples of these would include herpes simplex viruses, Epstein-Barr virus, which causes glandular fever. These are all diseases that once you're infected, the immune system doesn't eradicate the disease. Rather, what happens is it contains it. And so what that means is that reactivation of these viruses and relapses of the illness are indeed possible. And a really old observation is that, in fact, there is huge individual variability in the likelihood of these relapses occurring. So for some people, they occur relatively often, and for others, perhaps not at all. So as an undergraduate, when I was first introduced to this area, I was struck by the fact that a lot of the early research, so pre-1980s, had been fairly sort of primitive in the way that they'd explored what factors were determining who would experience a relapse versus who wouldn't. So a lot of the studies were really small. They typically had a retrospective design, and the assessment of what we would now call psychosocial influences was fairly imprecise. But in the 1980s, actually, the field was maturing quite rapidly. And what we were seeing was reasonably powered studies, prospective investigations, and the field had moved towards an approach whereby antibodies were being measured in response to these latent viral infections as a way of determining how well the immune system was containing the underlying disease. And a really important point to highlight about the way antibody was conceptualized in these studies is that essentially an increase in antibody was an indicator that the immune system hadn't controlled the underlying infection very well. And the rationale here is that the immune system, if it's failed to control the infection, it's enabled it to reactivate and replicate, and it's in response to that that you see an increase in antibody. This is really important because for the vaccine studies that I'll mention later, actually the reverse is true. So the greater the amount of antibody, the better the immune response. So important to bear in mind that increased antibody in the context of latent infections is indicative of a poorer immune response. And for me, one of the standout studies that was conducted in this decade was in fact by the Glazers in the mid-80s, where they recruited medical students, all of whom had some of these latent infections. And what they did was that they followed them up prospectively on 3 occasions, all of which would be characterized by different degrees of psychological stress. So they initially assessed them a month before their exams, again, just as the exam period was starting, and again at the end of a vacation period. And on each of these occasions, they measured antibodies specific to these latent viral infections. In a stroke of genius, they also measured loneliness in these students. And what they found was that the poorest immune response or the poorest control of these latent viral infections was observed in the loneliest students, and that the best or most effective control of the viruses, in other words, the lowest levels of antibody, were seen at the end of the vacation period. So in this single study, they signaled very clearly a potential role for both our perceptions of how lonely we feel, but also our experiences of stress as potentially being relevant to how the immune system is able to contain existing infections. And since that work in the mid-80s, of course, there have been very many studies that have replicated this and extended these associations. So we do have very robust data now showing that a range of different psychological and behavioral exposures appear to be associated with how well we control an existing infection. The flip side of this pincer movement was the excellent work of Sheldon Cohen and colleagues, these viral challenge studies. So here, very different to the latent viruses, what we're looking at is what happens when people are experimentally exposed to a new viral infection. Of course, the relevance to COVID-19 couldn't be greater. And the classic paradigm that Sheldon has used essentially involves recruiting a group of healthy volunteers who agree to be quarantined. This was in a time when most of us would have had no personal experience of what being quarantined means. But of course, I think most of us sadly know only too well what that feels like now. And during this period of quarantine, individuals would be exposed to one or more respiratory viruses. And what they would do is follow these people up to explore evidence of infection, which would be captured either by the presence of virus in the nasal cavity and or the presence of antibody, and critically also evidence of symptomatic illness. And again, this is really important because if we think about COVID-19, we know that a huge proportion of people who are infected are asymptomatic. So it's really interesting to think about what Sheldon Cohen's work has been able to illuminate in terms of this difference between being infected with a virus and for that infection to result in symptomatic illness. So again, a standout study, certainly for me, was one of Sheldon's early studies from '91, where they demonstrated a dose-response relationship between the experience of stress and the likelihood of developing a clinical cold. So the more stress you're experiencing, the greater the likelihood of developing a clinical cold. And much like the latent virus work, since that groundbreaking study, in fact, this observation has been repeated many times, and we've seen not only stress, but we've seen a range of health behaviors and also other psychosocial measures, such as measures of social support, all associated reliably and robustly with the likelihood of exposure to a virus resulting in infection and or illness. So that's to give you a flavor of what we knew in the 80s and what we've built on. But of course, the other thing that happened in the 1980s was that the public was confronted with a new public health threat. And at the time, most of us thought this is probably the worst thing that we can imagine. For those of you who can't remember, this, of course, was HIV infection. And I have to say, I think it's because of the pioneering work and this excellent research that was done in latent viral infections and also these viral challenge studies that it led investigators to necessarily take this same approach and say, could some of these associations that we've seen in these other contexts be relevant in the context of HIV? And I think what I'll do now is hand over to Steve, who can start unpacking some of what we learned as a consequence of the huge research effort that happened in that time.
Yeah, so I got interested in this topic about that time when we were trying to understand broadly why some people get sick and die quickly when they contract HIV infection and other people essentially would march on for a decade or more with no immune deterioration. And what was really surprising about HIV was how variable the course of infection was across individuals. And it took us collectively as a field, I'm certainly not the only person working in this area, but sort of Margaret Kemeny, actually a huge fraction of P&I researchers at one point in time became engaged in HIV research. And what I'll do is kind of roll up these sort of multiple strands of findings or evidence into a couple of major themes, not because they're necessarily, sort of historically that interesting, but simply because many of these themes turn out to be generic to human antiviral responses, and so probably pertain in one form or another to, sort of cold and flu challenge as much as they pertain to COVID-19 today. But what was unique about HIV was the degree of sort of, you could think of it as almost worldwide scientific interest in understanding this. This really defeated the optimism we had at the time that we had, vanquished infectious diseases largely. And as a result, there was intensive mapping of, how does the basic pathogenesis of HIV infection unfold? That was both important in terms of developing therapeutics and efforts to develop vaccines, but it was also important in giving us in biobehavioral research a really good roadmap at the cellular and molecular level for why do you end up with differences in, let's say, clinical disease versus asymptomatic infection? What's going on? What are the literal sort of gears and pulleys of human, both virological biology and human immune biology that end up creating differences in host resistance and survival versus disease and symptoms and that kind of stuff. So that made it easy for several groups to attack these kinds of questions, essentially using a sort of, you can think of it as like a similar concept of pincher activity or sort of top-down and bottom-up attacks, where you can start with essentially sort of risk factors that we've identified in behavioral research of one sort or another, things like loneliness or stress, and then kind of work down to, what do those things turn into in terms of neuroendocrine biology? How do they change cellular function, largely in the nervous system? What becomes, if you will, sort of the cellular and the biochemical representation in the body of these kinds of whole person experiences of social connection or stress or adversity, that kind of thing. At the same time, we can start with the viral pathogenesis work and move up from there and say, okay, given that we now understand in exquisite detail how the virus replicates, which cells it infects, how that causes disease, what causes, sort of long-term survival with health versus, sort of quick failure of immune responses, you can move from the kind of the molecular bottom up and try and essentially meet up these two lines of inquiry in the same place. And essentially what came out of this in a sort of a roll-up sense is that a lot of the things that are most, sort of recurrently identified as risk factors for viral vulnerability involve increased activation of the sympathetic nervous system, perhaps most sort of consistently, less consistently, but sort of sometimes observed are changes in hypothalamic pituitary adrenal axis activity. But in any case, those, you can think of them as biochemical representations of these risk factors. Then we could look at, essentially what happens when those chemicals are present in the vicinity of a viral infection or replication or immune responses to those. And within HIV, for example, we could reverse individual differences in clinical course essentially into individual differences in the rate at which they lost CD4 T cells. And that in turn, we could map back to individual differences in the rate at which the HIV virus itself replicated. And that in turn, we could reverse engineer one step further into variations in how well the immune system activates innate and adaptive antiviral responses. So what ended up happening is these two, is this sort of top-down line of inquiry and this bottom-up line of inquiry end up intersecting especially strongly in the realm of the earliest phase of innate antiviral responses that's structured by what we call type 1 interferon responses. Those are profoundly relevant, not just to HIV, but actually even more so to the SARS-2 coronavirus, similarly relevant to viral infections with flus and colds and that kind of thing. So I'll take a couple seconds to just sort of walk you through the roadmap of what these type 1 interferons do, what their role is, and how they connect to essentially everything else that happens in these kinds of antiviral responses. So I'm just going to share my screen here for a second and show you a little bit of a sort of cartoon version of how this stuff unfolds, or at least how it should unfold generically. So unfortunately, I can't quite tell if my slides are showing the way they should. There they go, that looks right. So in this scenario, essentially what we have is sort of, formerly healthy host cells now infected with a virus. And the first stage of an antiviral response actually involves not, the immune system or immune cells at all, but these virally infected cells detecting the presence of certain atypical RNA or DNA configurations in the cell, things like double-stranded RNA, which is highly characteristic of viruses, but rarely, if ever, found in healthy human cells. So we've got receptors that pick up that kind of thing, so-called pattern recognition receptors. And these pattern recognition receptors are essentially leashed by evolution to a large portfolio of genes called interferon response genes that can mount various forms of, you can think of as trip up and slow down responses within a virally infected cell. So these cells initially detect virus. There's really no, sort of conventional orthodox immune response yet, but already the cell has this capacity to detect an infection and slow it down by, let's say, changing its membrane structure, altering RNA transcription and protein production, and a wide variety of other things that if the cell pulls this maneuver, it will greatly reduce the capacity of this cell to be an effective viral production factory. That sometimes involves the release of type 1 interferons, particularly interferon alpha. There's about 23 different versions of that in the human genome, and interferon beta. Interferon beta is probably the key initiating event. And that has the effect not only of amplifying the cell's own innate antiviral response, but spreading locally to other nearby cells. And this is absolutely essential because it's the local nearby cells that will also be the primary targets of any virus that's released from this, kind of recently colonized now virus production factory. So essentially, the interferon system is racing for local containment of the local spread of the viral infection. That can also include, cells that get recruited into the environment by the destruction of a cell as it, produces so many viral particles that it literally falls apart. But the key event that takes place here is these interferons also play a very powerful role in recruiting into this environment and activating in that environment innate immune response cells, particularly dendritic cells, classical dendritic cells, plasmacytoid dendritic cells, macrophages, key cells from the essentially myeloid lineage of the immune system that recognize damaged cells and are informed by the presence of the interferons and other sort of antiviral response cytokines that the debris you see here didn't come from a cut, it didn't come from a bacteria, it didn't come from a worm, that, we see the fingerprints of a virus here. And so that leads these cells to not only swarm into this environment and start upregulating their own production of interferons and other antiviral control cytokines, but it also causes particularly the dendritic cells to what we call mature out of their nascent, you can think of as reconnaissance and patrolling form, and into a form where they will then not only orchestrate this local antiviral response, but some of these cells will leave that local environment, travel to the lymph nodes, and start to interact with other cells of the immune system. Essentially, this is the bridge to adaptive immunity. This is when the information in the infected tissue now is getting delivered to the immune system's central command and control systems as these dendritic cells, predominantly to a lesser extent macrophages, become antigen-presenting cells. They start to show T cells the kinds of debris they found in the environment of these damaged cells. And that is essentially sort of the physical calling card for what kind of a response the immune system is going to need to make. So that presentation of viral antigens to T cells kicks off both the production of T cell responses that which I'll talk more about in a moment, as well as the production of B cell mediated antibody responses. So these antibodies then get synthesized, circulate through the blood, and are capable of intercepting viral particles as they leave the factory that produced them, the producer cells, and before they infect any of their neighboring cells or cells elsewhere in the body. The difficulty with antibodies, though, is that they are big proteins that are not capable of actually getting inside the cell and stopping the viruses. The viruses, because they're inside the cell, are largely shielded at that stage of their existence from attack by antibodies. So we need a different response that involves cytotoxic T cells that also get made in this lymph node environment. And they, unlike antibodies, can peer inside into the guts of a cell and determine whether the cell is infected and kind of actively producing new viral proteins. And so these cytotoxic T cells will sort of deliver, ideally at least, the second punch, which is they'll recognize infected cells and essentially kill those cells. They essentially activate suicide programs on infected cells and stop them from being productive virus factories. None of these responses, particularly the antigen presentation, the antibody production, or the cytotoxic T cell production, none of these operate very efficiently unless there is good signaling out of the local tissue environment in the first place from the interferon system to kind of kick off all of these, both these more sweeping localized innate responses and these more systemic and ultimately powerful adaptive immune responses from antibodies and cytotoxic T cells. So this scrimmage over between viruses and the innate antiviral response turns out to make all the difference in the world. Even today with coronavirus infection, the biggest single genetic influence we can find over who gets, sort of badly sick and dies versus who basically contains these infections while they're still asymptomatic and doesn't even know that they've been infected. The single most reliable predictor is how strong is your complement of genetic polymorphisms in enabling this innate antiviral response, this production of type 1 interferons in the initial stages of infection. If the type 1 interferons can outflank the nascent viral infection, then you may well never have any symptoms at all, even though your body has kind of contained this pretty well. Now, remember the top-down, bottom-up story. There's all kinds of molecules and cells involved in this, sort of array of responses we talked about. But the single quantitatively most vulnerable point in this whole ecosystem is actually the production of those interferons in the first place. Having, sort of stress levels of norepinephrine or stress levels of cortisol in the local environment of these infected cells will knock that interferon response down by 80% or more. So it's not a trivial effect. It's a really, really big quantitative effect. And there's lots of other stages in this system where, sort of this stress endocrinology and stress neurobiology can amplify the cytotoxic T cell resistance presenting cells not to make so much interleukin 12. And so you don't get a very strong so-called Th1 response and cytotoxic T cell response. Another place where there's a lot of vulnerability involves the sort of the battleground that's created by the immune system's interaction with virally infected tissue. So as the immune system starts killing cells, or as they die due to the endogenous damage from viral infection, you get inflammation. And we now know from work, actually this was beginning to be quite evident in Sheldon's initial studies, where they were finding that nasal fluids contained more pro-inflammatory cytokines in the people who were getting sick, and that tended to be the people that had more stress and less social connection and that sort of thing. And that theme recurred in HIV, where inflammatory biology is one of the chief fuels for HIV replication at the cellular level. And we now see this again in coronavirus infection. What defines COVID, the disease, isn't these antiviral responses. It's, do you get a huge wholesale inflammatory destruction of airway tissue that essentially is a consequence of the viral battle, not typically intrinsic to it per se? And can the body, prevent that inflammation from becoming so extreme as to prevent respiration? So these intersections between what the nervous system essentially has kind of been instructed by evolution to tell the immune system to do, and the sort of the quirks and idiosyncrasies of these viral infections, ends up being quite influential in structuring, how badly disease arises and how far around the curve you get before we can kind of, contain it. these responses are not only relevant to the question, I'll call it the kind of the CAV1 and Sheldon question about who gets sick in the first place? How big does the infection get before it finally gets corralled by cytotoxic T cells and antibodies? But it also has big implications for vaccine-induced immune responses and the ability to mount a primary response that will hopefully contain these kinds of effects in any kind of future exposure. Particularly the role of antigen-presenting cells, absolutely essential in kicking off productive antibody responses, productive cytotoxic T cell responses in advance of a natural infection. So if the immune system is working less efficiently because the norepinephrine or the cortisol is essentially informing it, make less efficient, don't activate as much, don't produce as much interferon, don't send as much signal back to the lymph node about what kind of an immune response we'd like to create as a function of the presence of these antigens in the local environment. That makes all the difference in the world. So that becomes kind of I guess the best way to put it is a linchpin event in whether we'll even be able to generate a strong immune response by vaccination in advance of natural infection. For those of you who are interested in more of both the kind of the psychobiological and the sort of psychoneuroendocrine mechanisms, actually there's great recent reviews on these topics from Jan Glazer, most recently speaking kind of in the direction of vaccines, as well as Sheldon Cohen, who wrote a great review early in the COVID crisis around, sort of what do we know from the flu studies and what can we kind of project is going to hold in coronavirus infection. I can tell you he's spot on so far based on what we've seen. So this strand of thinking that your psychology matters for your antiviral biology seems kind of grim if you think about it from a kind of a stress perspective, but it also opens up interesting opportunities for intervention. Like if we can change people's psychology and psychobiology, can we perhaps enhance, let's say, the vigor of vaccine-induced immune responses? And that optimization opportunity, I think, is really an interesting, sort of point at this moment in time. And this strand of thinking, I think, is particularly, sort of exciting at the moment. And one of the people who's actually worked in this area most extensively is Cav. So I'm just going to hand it back to Cav and have her give you a little update on what they found in general and what the prospects might be for coronavirus vaccines now.
Thank you, Steve. So I'm going to go a little bit back in time first before I touch on some of the more recent intervention evidence, if that's okay. So in thinking about vaccines, what I do want to do is to bring you back to the 1990s, a very important decade. For those of you who are struggling to remember what happened in the 90s, it was the time that the first Harry Potter novel was published. It was also the decade that gave us the Fox News channel, which I think we could have a whole other meeting about. And from a behavioral science perspective, I think it was a really important period because for me personally, I think that there was a really important paradigm shift in how we are exploring and trying to understand this relationship between behavioral factors and viral infections. Really, what I'm hinting at is the growth in what I like to think of as the vaccine challenge studies. So thinking back to what I was saying earlier, in terms of the viral challenge studies, these studies were designed with the very explicit aim of eliciting infection and seeing how well the immune system responded to that in terms of protecting us from that. But here we've got the vaccine challenge studies where the very explicit aim is to see, well, do psychological and behavioral factors have any impact on how well vaccines will work in protecting us from these viral challenges? The typical paradigm was and continues to be one in which scientists will focus on a population characterized by a particular psychological or behavioral exposure. So it could be acute or chronic stress, it could be a behavior, it could be a measure of social support. All of these have been investigated in the last 20 years or so. And what happens is individuals are given a vaccination. Most commonly, it's a standard dose of vaccine. And antibody levels, again, the focus, Steve has talked a lot about the T cell response, which is hugely important. But in these studies, primarily, the focus has been on the antibody response. And investigators have been interested to see how the antibody response to the vaccination is potentially attenuated or enhanced, depending upon the study design, by these psychological and behavioral exposures. For me, one of the standout studies within this area was again by Jan Kiecock Glaser and Ron Glaser, where they very explicitly set out to ask the question, did the combination of being an older adult as well as being chronically stressed put you at greater risk of influenza vaccine not working? And with this study, as with lots of the others, they used antibody levels as their indicator of how well the vaccine was working. They focused on spousal carers of dementia patients as their chronically stressed older population and age and gender match controls. All of their participants were given a flu vaccine, and they looked at the proportion of people in each group who were able to produce a clinically appropriate response to the vaccination, which in the context of flu is traditionally denoted by a fourfold increase in antibody. And what their results showed, and I personally found this very striking at the time, was that although 2/3 of the control group produced this clinically appropriate response to vaccination, only about 1/3 of the chronically stressed caregivers did. And I think one of the reasons why these results really struck me was because what these data were essentially saying was that the effects of chronic stress on the immune system of older adults were sufficiently profound that potentially for up to 2/3 of adults, they were rendering flu vaccine completely ineffective. That's hugely important from a public health perspective because, as I'm sure you appreciate, flu in older adults is a very serious illness. It uses up a lot of healthcare resources, and of course, it can be fatal. So we had here the beginnings of some evidence which I think really pushed this body of work up the agenda, certainly in my mind, because it was really signaling that not only were these really robust relationships scientifically, but these relationships were potentially clinically relevant. And of course, what we've seen since that work in the 90s, in the last couple of decades, is this pattern of findings between a range of different psychological and social and behavioral exposures and alterations in how well vaccines work. And several really good systematic reviews attest to this. I don't think it's any surprise then that what we've also seen is a growth in intervention research, clinical trials, where people are essentially asking, well, can we harness these influences? Because after all, these are modifiable factors in theory. Unlike your age, you could potentially modify your stress. You could potentially change your diet. You could potentially change your physical activity. And in so doing, could we optimize the effectiveness of vaccines? So for my sins, I thought a few years ago that it would be really useful to try and synthesize all of this evidence. And my motivation really was to try and say, well, is there one intervention approach that's emerging amongst all others that seems like it's going to give us the biggest bang for our buck, if you like? And I have to say, it's a review that has taken us several years to complete because this area of research has been growing really rapidly. The review itself has been published really recently in the last few months. And what I can tell you is that actually in that relatively short period of time, we've already got over 100 clinical trials in this area. What's really interesting, though, is that the vast majority of these trials are dietary based. Relatively few look at exercise-based interventions, and even fewer are considering psychological interventions. So straight away, to me, and I hope to many of you, that signals that this is an area that is ripe for us to be getting immersed in, particularly in the context of COVID-19 and the context of COVID-19 vaccines. In terms of the take-home messages of the review, what we can say is that in keeping with this being a nascent area of research, we've got early signal that these non-pharmacological approaches, if you like, hold some potential. They do look like they might be a vehicle by which we can optimize vaccines that otherwise don't work well. And I think it's worth saying that in the context of COVID-19, certainly 2 out of the 3 vaccines that are licensed here in the UK the clinical trial data might suggest to you, well, 95% effectiveness doesn't look like there's much room for behavioral science here. But it's important to remember that in the real world, vaccines typically don't work as well as they do in these clinical trials. And I don't know if you've seen it, but the data out this week from Israel, where they've vaccinated a really large proportion of their population, is already signaling that COVID-19 vaccines are not working as well as these clinical trials have suggested. So I do think that this is an area where there is actually a really urgent need for us to collate what we already know about psychological and behavioral influences on viral disease, but also vaccinations, and explore ways in which we can develop rapidly interventions that are going to be fit for purpose for this context, bearing in mind that what we're looking at is an unprecedented public health problem where we need to intervene rapidly and quickly. So this talk of vaccine, I think, necessarily brings me to Heidi. And for me, I think Heidi's work is so important in this area because notwithstanding all of the research that Steve and I have talked about, fundamentally, if people don't agree to have a vaccination, then all bets are off. So I'm going to hand over to Heidi, and she's going to talk about some of her excellent work in this area.
Thanks so much, Kavita. It's been, I couldn't think of a better, how do you say, entry point for what I was going to talk about. And in fact, I wasn't going to show slides, but between your and Steve's presentations, I've added a few to one map, which I was about to put up. My work, I lead the Vaccine Confidence Project at the London School of Hygiene and Tropical Medicine. I'm also have a co-appointment at the Institute of Health Metrics and Evaluation at University of Washington. And basically, I'm trained as an anthropologist. I've always worked in health, but looking at some of the social cultural behavioral factors that affect uptake of health interventions. But I led UNICEF's introduction of new vaccines and strategy and communication in 2000, 2005, and saw this growing epidemic of questioning around vaccines. But the big question people said to me was, well, you're being too negative, focus on the positive. And everybody just kept asking me, how much has it gotten worse? And I said, we don't have a benchmark. We can't compare it. We don't have a metric. We can't measure it. So I decided to establish the Vaccine Confidence Project to try to bring some metrics around this messy emotional space of vaccine confidence and vaccine emotions, because I realized that until I started to put some numbers on it, nobody was really going to pay attention. It has dramatically changed. The public attention to it. And we developed a vaccine confidence index in early 2000. 2014, 15. And I'm going to start by showing you our relatively recent publication in The Lancet, which looks at the last 5 years of public confidence in vaccines to show you the landscape. So our Vaccine Confidence Index, which was a bit modeled after the Consumer Confidence Index, and I like the term confidence way more than hesitancy because, one, it doesn't put judgment on it. You can have 0% confidence or 100% confidence. But also, like the consumer confidence is an indicator of your willingness to, in the consumer one, willingness to spend, in this case, on vaccines. Now, the darkest red is the least confident, and the darkest blue is the most confident. And we have 4 domains of confidence we measure: confidence in the importance of vaccines, in their safety, in their effectiveness, do they work, and in compatibility with religious beliefs, because those were 4 key areas after going through many batteries of much longer questionnaires. These 4 were pretty crucial. Now, this one is just the map in the Lancet of comparing 2015 metric on vaccines are safe with 2018. We also have maps on effectiveness and importance. But the safety one to me tells a story. Europe, we identified in our first global study on vaccine confidence, which was in 2015. It was in eBiomedicine. Europe turned out to be the most skeptical, vaccine skeptical region in the world, which was a bit surprising coming from the US. I was sure that, but I think sure that it was there. But I think if you disaggregated the US into states, you would see some states looking a bit more like France, which turned out to be the most skeptical country in the world, with only 43% of the population in a nationally representative sample saying that they thought vaccines were safe. 43% saying they did not think vaccines were safe. So the EU, the EU asked us to be monitoring over time, and we've done that also with the rest of the world. And they have made a concerted effort around this issue, and you can start to see that they're lightening in their color. But what's very concerning, and another thing that has been revealing in our work, and we look at trends over time, if you look at Africa on the left, which is dominantly blue, more on the positive end, it's become more yellow. And this is specifically in the Francophone countries. And this is reflective of what we're seeing as not how sentiment is spreading with language diaspora. Poland has a huge anti-vaccine, highly organized group. Wakefield's visited there a few times. And we are now seeing in Scotland and England growing hesitancy in the Polish communities to vaccinate. So these language diasporas have an influence. Now, coming to our more recent studies, which we've looking more and more at COVID vaccine, you can see equally here, it is highly variable. There's very few countries in the world that are the same in their answers on willingness to take a COVID vaccine. Lebanon is the strongest with definitely will not. And so much of this is also embedded in trust in government, trust in the system. We have done a lot of other questions around this. And for those, we find a very strong, the highest correlation, more than age, sex, and other demographic factors. Public confidence, if the handled the COVID response well, the more likely they are to take a vaccine. So these are external factors, but there's also internal factors. And some of the other external factors are the what in my book stuck, the epidemic, the infodemic, as WHO calls it, of misinformation. I also, in this case, I have a lot of media and other questions. Okay, there's a lot of misinformation. Does it really affect people's willingness to take a vaccine? So we did a control trial. We took 8,000 people, 4,000 in the US, 4,000 in the UK, interviewed them all about their experience around COVID, as well as willingness to take a COVID vaccine. And then 1,000 of them, we gave very straightforward factual information about the COVID vaccine. For 3,000 in the US and 3,000 in the UK, we showed them one at a time the most frequently circulating pieces of misinformation, and then interviewed everyone again around COVID experience and willingness to take a COVID vaccine. In the UK, 54% had said they would definitely accept a vaccine before the exposure, and it dropped 6.4 points to 47.6, having seen those pieces of information. And among those 5 pieces of information in both the US and the UK, it was the ones that had the most scientific look to them that had the most significance, particularly the mRNA vaccine one that was looked like it was from a genomics textbook, but the bottom line was first they feed us GMO and now they're trying to turn us into GMH, genetically modified humans. It may sound crazy, but I think we all know people believe all kinds of things. This is my book, which has a lot more background on this, but this whole issue of, and I've been listening eagerly to what both Kavita and Steve, but particularly this issue of emotions and everything from a whole spectrum of emotions and psychological issues, not just issues, states, because the book talks about rumors, but really everything in here, when I hear about latent and I hear about reaction, one of my premises in Stuck is that rumors are thriving, particularly in this context with COVID, of a landscape of uncertainty, but they don't go away, and they kind of hang out in the background, and individuals, communities, and countries have historic memories that affect people's willingness to take a vaccine. It is, the old story, if you've been bit by a dog, the next time you see a dog, you react. I mean, everything we've been hearing about in the previous discussion is about how, there is an internalization, not just individual immune systems and vulnerability, but there's at the community level, you have these. One of the reasons I've particularly gotten fascinated by, go back to the latency of rumors, that you have these backgrounds and you can think that you're clipping off a piece of information or putting a rumor to bed, but if something comes along that can spark it back, it'll be back. It doesn't go away, particularly in settings where there is a history of distrust or a bad previous experience. One of the other areas that has gotten me particularly fascinated and studying, because it has serious public health impacts, is the relationship with anxiety and stress on actually provoking adverse events following immunization. It's interesting to look at on the one hand, it affects effectiveness of how you take the vaccine. I mean, the effectiveness of the vaccine. But on the other hand, another aspect of the anxiety and underlying emotional states and psychological states is in this whole area of adverse events following immunization. The World Health Organization has what they call a Global Advisory Committee on Vaccine Safety. And for years, I've been trying to say it should be vaccination safety, because it's not just what's in the vaccine, it's the vaccination experience. That is, I mean, when I was at UNICEF, we saw a number of child deaths because of issues with inhuman error. And where does that fit in? And how can you just say it wasn't caused by the vaccine when in fact it was the experience of immunization and the human error? But in other cases, we have seen clusters, for instance, particularly around HPV, clusters of girls fainting, neurological issues, shaking, long-term issues in a number of countries. And it's one thing about this social media environment we're in, we see what I call in stuck a contagion of emotion. We see 12 girls fainting paralysis in Japan after HPV. YouTubes go to Denmark. We have a large group there. It gets translated into Spanish, voiceover in English, and original video in Japanese showing up in Colombia. 600 girls across multiple schools panic and all start having psychosomatic reactions after vaccines. This has become such a serious and repeated phenomena, particularly amplified in the context of social media, that the World Health Organization is now included in their Global Advisory Committee on Vaccine Safety new guidance acknowledging these, what they're calling stress-related responses, because as Kavita was talking about, can you modulate some of these? If you can calm people before vaccinations, when you know that there's this vulnerability, will it mean that one, they'll accept it and will less likely to have an adverse reaction? Sorry, I didn't mean to go on so long on this, but it is a huge area and an issue that has been understudied. And I'm very concerned about the context of COVID, because I think we have whole populations that are extremely stressed in many ways, and how that will affect COVID effectiveness, but also the potential, particularly in mass campaigns, for these kind of stress-related responses after vaccination. And the last slide, and I'd be really keen if anyone was interested in contacting me about it, we launched with the Lancet a commission on the emotional determinants of health. I was particularly interested in this because of the areas that I just mentioned that we've been looking at. But COVID hit just after we launched it, so we're still going to move forward on it, but we're still putting together the commission, so I would welcome anyone to come back to me on that. Thanks so much. And sorry, that's a bit of a different, trying to bring some of the elements, but a bit of a different perspective on the, from the vaccine and emotions and histories point of view. Thanks.
Heidi, thank you so much. That was absolutely fascinating and just of critical importance. We're going to have a few minutes of our three panelists asking each other questions and having discussion, and then we're going to open it up to all of the rest. We have so many experts with us today in our audience, so I'm very excited to hear the discussion. I suggest that you type in your questions and then I can call on you. So you could type them in now while the panelists are talking. Thank you.
May I start, Melissa, if that's okay? Heidi, I've followed your work and I think it's so important, and the timeliness of it couldn't be greater. I'm just wondering, from what I understand, actually, people's responses to vaccination can vary between vaccines. So it's not if I'm right, I think it's not that, it's not a trait per se. And I was just wondering, are there characteristics of this pandemic, or indeed of this disease, that you think are going to make the issue of vaccine confidence a bigger problem than we have seen it historically?
Well, I think one of the challenges with the current vaccines is particularly the mRNA one, which is Moderna and the Pfizer ones. This is brand new, and the public does not like things that are brand new. They don't like things made fast. They meaning one of our biggest challenges around H1N1 in 2009 was people not wanting to take the H1N1 vaccine because they thought it was made too fast. And this is a virus we know well. It's even a strain that we had historic experiences with, and the vaccine was not particularly atypical. It was just, it hadn't been identified early enough to get into the seasonal flu vaccine, so it was separate. Even then, people were like, made too fast. So one of the biggest angsts is the fast, the speed and the newness. And will there be, I mean, there has been a lot of things circulating in social media about, is this going to be another thalidomide story? And so that's one issue that's going to is affecting people. I think also the state of the pandemic. People are constantly, we've been monitoring frequently since March, April, people's willingness to take a COVID vaccine. And barely anyone was talking about it in March, April. There it was like mask, no mask, 3 feet, 5 feet away. It was all pretty confused scramble to figure out how do we control this. And people said, when asked, would you take a vaccine? It's like more people said yes then in any of the country studies than they do now. And I think it's because they know more. And also it's here. Then it was like something in the future. Yeah, sure, I'd take a vaccine. This is big and scary. But also knowing that it's like down the pipeline, I don't have to worry about it yet. And so we've also seen, in the summer, the willingness went down a bit, partly because they felt less threatened. Then it starts to go up a bit. So people do, this whole cycle of what has been historically separated as reason and emotion. They're all, as you know well, they're not separate. But people do weigh in a number of things in their environment. And particularly, there was a beautiful piece by Danielle Offrey in the New England Journal of Medicine in around the 2009 epidemic. She called it emotional epidemiology. And she's a GP, and she was talking about how the emotions of her patients at different points of the pandemic or emerging pandemic, in terms of their willingness and really wanting it, when are we going to have it, to I don't think I want that thing, to it was fascinating.
Wonderful. I'm going to combine several of these fantastic questions that are coming in, and they're about implication and application, and particularly for the health disparities that we see already and for reducing health disparities. So to Heidi, thinking about we understand that what is predictors of the vaccination hesitancy or lack of confidence, trust, and the cultural bubbles and language diasporas these transfer in. How do you best counter that social media rapid transmission? So scientific information alone, we know, can polarize people who are already feeling disbelief or anti-vax attitudes. And then the other application question for Kav and Steve is, what do we do at the time of vaccination, the week before vaccination? What do you suggest psychologically, behaviorally, sleep-wise, nutritionally? We have Bonnie Kaplan with us today, one of the experts in nutrition and psychological health and psychiatric disorders and the immune system. So let's start with Heidi. What do we do with this? What is your best guess on what a social campaign would look like, or a heavier touch intervention?
I think one of the main reasons we set up our kind of early listening, and I showed you the survey work, but we're doing, we've kind of grown with the landscape of social media. So we're doing a lot of 24/7 listening. But there are some like longer term, deeper things like the trust in cultural and historic experiences. And what we're trying to do is look for early signals of issues so that we can get in there as soon as we can with, you don't want to get confrontational with some of these anti-sentiments. And when there's a weak spot in the system, it's a group that is distrusting or whatever, getting in there, engaging, also remembering that the world is not just on social media, that they actually talk to each other. In some cases, I think sometimes we forget that, and try to instead create a surround sound, as it were, on social media to try to rein it in. I work closely with a lot of social media companies. I have mixed views about shutting all of it down, because it doesn't, clipping off the bad stuff isn't shutting down the emotions behind it. And I think we're facing right now quite a challenge, because on the one hand, I totally get and have, say, a breath of relief when things are shut down, especially when you against more toxic narratives, but it does mean it's harder to find them and rein them in. So it's a constant, basically these days, we need to be constantly on top of it and try to get ahead of it. But it's a challenge. It's a good question. There's no easy answer.
In the climate work, they've shown that it's not just scientific facts, those can backfire, but rather emotional appeals and narratives that are
Yeah, but it's not really one or the other, because we did see with that misinformation study that actually it was the science-y thing. So you want to have some level of credibility in it, but make it, humanize it. I think we tend to say science or emotion, but actually we want science with emotion.
Thank you. Cav and Steve?
Steve, shall I go first? Yeah. Okay. So the question was around what would we suggest would be appropriate psychological or behavioral things that we might do. So drawing upon various bits of evidence that I can think of. So firstly, from our review, the strongest data that exists as a result, I think purely because of the number of trials that have been done, is in terms of dietary interventions. And there we see that the use of probiotics seems to have an early signal of benefit. So if you're going to do anything dietary, I would suggest probiotics based on our recently published review. Based on another study that we did a few years ago, which was an observational study, it wasn't an intervention study. And what we did in there was we put head to head a range of psychological and behavioral exposures that had previously been associated with how well, in this context, flu vaccine works. So we looked at physical activity, we looked at positive and negative mood, we looked at sleep and a range of things. And in that study, certainly, when you put them all into the model, interestingly, the most prominent predictor of how much antibody you produced was positive mood on the day of vaccination. So on the day that you get vaccinated, find your funny, do something that makes you positive and puts a smile on your face. The other things that there is evidence for, again, mostly from observational studies, is not smoking, moderate alcohol intake from Sheldon Cohen's work. I mean, I'm sure Sheldon would agree with this. In his studies, people were typically really healthy, so he didn't have the really heavy drinkers. But in terms of the moderate drinking, that says to me it's okay for me to have a glass of wine maybe the day before, but that might be something just about me. And I think a really under considered area, which I'm quite fascinated about, and I know Greg Miller's done some stuff in this area, is thinking about the days after the vaccination. So, of course, we know that the immune response isn't instant, which is why it's really critical that people don't get a vaccination and then instantly, forget about all of those protective behaviors we need to engage in. It's a process of 2 to 3 weeks at least for that first boost. So, what are the things that we can think about in terms of our emotional health and physical well-being in perhaps that week immediately after a vaccination to keep us on an even keel? Those would be my top tips.
Beautiful. And Steve, those are so important. Thank you. And I think just so relevant, particularly in the next few months. Steve, the idea of the stress hormone response modulating the interferon and eventually antibody response, can you add to your answer how to think about acute stress helping the vaccination response, some of Firdousa's work, or the acute immune response, versus this great suggestion in our chat, which is, shouldn't we be doing diaphragmatic breathing, right before the vaccination? Thanks.
Very possible. There's actually a nice array of findings connecting respiratory sinus arrhythmia to better interferon biology in general. Nobody's actually done this kind of thing as an experiment with a vaccination, but that actually is, based on the data available right now, actually quite a plausible way to go, to try and sort of like by practices goose the parasympathetic nervous system to try and push aside this sort of particularly sympathetic interference with optimal responses. And apropos Cav's comment, it might not be a bad idea to keep doing that periodically over a few days, because vaccines are unfolding events. They're not just one time. We experience them as like it happened now and it's done, but the reality is these are immune responses that are getting constructed over literally a week or two. And so thinking about the post-injection phase as a unrecognized opportunity. But I would say, in addition, I generally worry about the ultimate impact of these parachute interventions, where you leave the catastrophe that is the rest of life and you just say, I'm going to parachute in and fix this day, and then your biology is going to be okay. The reality is, that's always going to be hard compared to actually trying to fix the broader themes of our current existence. And I think one of the themes that comes through in Heidi's book is that a lot of vaccine, lack of confidence or reactivity comes not because of the vaccine itself, but because of vaccination as a symbol of bigger issues related to my trust in my fellow human being, my trust in my government, my trust in science and technology, for which there's, all of which have more than a grain of truth and legitimacy to them. So I think, to bring it way back to the beginning, what we have failed spectacularly to do in current public health interventions, in my opinion, is learn the fundamental lessons that we actually learned a couple decades ago in HIV. And one of the lessons was that centralized, governmental, paternalistic, public health or medical advice is generally not terrifically effective as a persuasive campaign. When we turned the corner with the HIV epidemic is when we stopped trying to get centralized government ministers to scare people or cajole people into doing things, and instead turned to community-based organizations. So to be really concrete about this, when AIDS Project Los Angeles or the San Francisco AIDS Foundation or Gay Men's Health Alliance, when they're telling you, we think the vaccine is okay, we think you should do this, we think this actually makes sense, and we're from your community. We're not for, pharma profits. We're not for the government, telling everybody what a great job they're doing. We're not for pretending that vaccines never cause significant adverse events. We're for, in the HIV example, gay men's health and survival. And we think, all things considered, given everything in your life, our advice is this is probably worth the risk. Those ultimately are much more credible and much more impactful. But I see just the opposite of that going on in our health messaging right now. So I think tragically, it's not going to be until community-based organizations that are trustworthy for the individuals who live in those communities will be able to kind of endorse this kind of stuff. And that is also the source of the confidence and the security and the kind of the social relational well-being that actually tamps down a lot of this chronic stress biology in the first place. So I think we're missing huge opportunities for impact in terms of overlooking trusted communities, trusted community members, and these very localized intervention strategies as opposed to these big top-down, implicitly governmental and paternalistic approaches. Those are fine for the populations that respond well to that kind of stuff. But I can tell you in all major developed economies, you're going to be missing a third to possibly 2/3 of the population if that's the sole approach you take.
Okay, so we shouldn't acutely stress ourselves right before we get the vaccination to increase.
I would not recommend it, but I don't think it's going to make that much of a difference in the first place. I think where the acute stress, generically, no, I would say. But it's worth paying attention to the issue that Heidi brought up in terms of anxiety responses. So, anything that you do that primes the body for more, cardiovascular reactivities or, sort of other kinds of essentially autonomic responses, that kind of stuff, I think worry, is just going to load up actually the risk for these localized anxiety somatic responses that then, as Heidi articulated quite well, go viral when people represent them. You know, within the first 2 days of Moderna vaccination in the US, there was a woman on YouTube saying, I've got Bell's palsy because of this. And she's right. That's almost certainly true. But to try and bury that, you know, it's not like people don't know that. People will, this will get around. So pretending it doesn't happen or saying that's not true or that's not important, just not going to be a productive strategy.
Thank you. One of the questions from John, we have several Johns, so I'm not sure which one that is, but durability of antibody response. Does that differ by chronic stress? Should we expect and worry about BIPOC communities? And the question was in particular was, what about the healthcare personnel who are going to be more exposed to virus? Are they going to have a lesser antibody sustainability? John Sheridan. John, did you want to explain? I think maybe you could explain your question better.
Unmute, John.
Hey, Steve. Can you hear me?
Yep.
Yeah, okay. I'm concerned about the chronic nature of what our healthcare workers are going through. The mental anguish that's expressed by especially our nursing corps has just been remarkable. So here we have a group that's chronically stressed, and folks have mentioned Jan Kiko Glazer, Ron Glazer's studies before. This is a group chronically stressed over a significantly long period with very little relief in sight. And now we're vaccinating them, and they're the first ones to get the vaccines. So I have a concern about whether or not it's going to be efficient in that group of individuals compared to the more general public.
My sense is that's probably true. I think the pragmatic question is just like, what other course of action do we have? I mean, do you want them all to meditate? That'd be awesome. But I mean, seriously, I think that that's a real risk and a real concern. I'm not sure, you know, at this point, what we would do to head that off. But it does bring up a really interesting question. And this is something that we've kind of actually evaded. And that is, if we, and we're not at this place today, but if we keep pecking away at this mechanistic research on exactly how in the cellular and molecular level this kind of stress biology interferes with vaccine-induced responses, it shouldn't actually in principle be impossible to do what we've started to do in cancer, which is like, okay, if you're going in for cancer surgery, it's not a bad idea to load up on beta blockers for about 2 or 3 days before that. That looks, and you know, you don't need to park on beta blockers the rest of your life. You just need to kind of taper off over the next week or so. So the kind of the Shamgar model of, if you've got a really pivotal biological event and you know that the stress biology catalyzes adverse biological consequences from, in this case, surgery to remove a tumor, it might not be crazy to pre-medicate. Now, again, you know, obviously we're not there today on exactly how you would pre-medicate to maximize vaccine responses, but it does bring up interesting questions about not only pre-medicating a body to be ready to respond, but actually including in the adjuvant pool things that will locally modify stress biology. Most of the time when vaccine manufacturers talk about adjuvants, they're like, how can I make the, you know, sort of the antigenic property or the local inflammatory immune antigen presenting cell driving response stronger? But you could potentially go in there and also say, how can I inhibit the stress biology in this local environment? In which case you don't have to even systemically perturb a person. You can just kind of throttle down local biases.
Thank you so much. I want to make sure we have time for our community discussion. And before we do that, I would like to, well, first of all, just thank Heidi, Cav, and Steve for phenomenal information, and just your life's work is, you know, so important, and applications, and they all have applications. So we are, as a community, are going to be using this information professionally as well as personally. Many people in our group of 100 behavioral medicine scientists here are leading COVID or very involved in the different COVID movements, both at their universities, at their states, and nationally. Before we close this section, I would like to invite Nancy Klimas. Some of our we have people who have devoted their lives to the study of stress and immune response, et cetera. And I would love to hear from you. So if you would like to comment or ask a question, you know, Jan and Sheldon, and you know who you are, please join us.
Nice to be included in that August list. That's fantastic. Thank you.
Thank you for your great comments. I thought we could, you know,
about the risk of post-COVID disease and what the role of secondary immunology has to do with the people who are put at risk for long haulers syndrome or post-COVID sequela. And the other thing is that there's evidence now of very significant subclinical organ damage post-COVID, even in mild disease, with lung and heart being the most obvious, with cardiac fibrosis on MRIs and lung fibrosis and people who were subclinical. So these are all places where the role of the inflammatory immune system and the immune response have a lot to do with the risk of long-term consequence to the acute infection. And I put in the chat that there's $1.15 billion of new NIH money to understand this better. So all you people with your grant writing pens out.
so much, Nancy. I see Sheldon has appeared.
Yeah. I just wanted to make one point about stressors that I think is particularly important to COVID. And one of the things we found historically in our work is that there are really two kinds of stressors that are most potent as predictors of susceptibility. And one is interpersonal stressors, and the other is economic stressors, being unemployed or underemployed. And these are obviously central issues in COVID, and making this work, I think, even at least giving us evidence this work is even more relevant.
Yes.
Heidi, Steve, and Cav, did you want to add any last comments before we move into our membership coffee time?
No, it's but my husband had very serious COVID and long COVID, and and I've been reading a lot, and my stepmother got COVID from the care worker who came to take her temperature. So I've been following pretty close to home a lot of and one of the things that I mean, things like there's immediate things like loss of hair and things that they're relating less to the COVID virus and more to the the trauma of being on oxygen in a hospital, in isolation, with no family, nothing, no communication, no extended periods of time. And as you said, the healthcare workers are going through even more. Aside from the viral specific, I mean, one of the things I've seen is they're trying to disaggregate what's actually a sequelae of the virus and what's actually a sequelae of the experience of the virus. And just that to me is a fascinating distinction. Not that it shouldn't be a candidate for the 1 whatever billion. I think just it's another topic of research to look into that.
I think we're in a weird space right here, right now, and all of us as healthcare professionals and investigators need to realize how much the entire world is leaning into social media to inform them what to do right now before there's time to write or get data. And we all know how long it takes to get data, but at some point you have to just trust your instincts as a scientist and very knowledgeable in the field and give good advice. And when it comes from a group like this, it's extremely helpful. I mean, putting out a guidelines or some advice or a YouTube video that came from ABMR would be, I think, very helpful right now, just to let people understand the general principles and how they can help themselves. The role of cholinergic excess right now, what you're just describing, Heidi, and hair loss. I mean, that's just, small. there's a lot of mechanisms there, but one of them is, you know, shutting down all those tiny little vessels that infuse the nutrients into hair follicles. That's cholinergic excess. You know, that's a stress response with a biologic implications. We're seeing it. And these post-COVID people who are pushing out into long haulers have a lot of autonomic symptoms, just loads of them. I think we can do a lot with what we know about reinforcing parasympathetic in a very practical way and lay that out there. They're not going to hurt themselves. And if we wait, you know, a year and a half to give the advice, we're waiting way too long.
Yeah, thank you, Nancy. Ian Cronish just added, he's linking, their group's linking PTSD symptoms to COVID long haul symptoms. We probably, Roxy and Allison Holman probably have data on this too, because they've been studying at a national level, the mental health symptoms. So we are going to turn to John Ruiz and